Okay, so we talked about the structure of membranes, and now we have to talk about movement of things across. That's what mainly membranes do, is move things in and out of the cell and in and out of organelles. And so, like we talked about, membranes are semi-permeable, so they only let certain things through, only let certain things across, and that permeability is controlled by the proteins, by the transport proteins that are in the membrane, and those proteins are controlled by the cell's DNA, by what DNA is expressed, what DNA is read. So, what are the kind of things that move across a semi-permeable membrane? Well, we've got water is a big one. So cells have to balance the amount of water that's in them. So there are channels, aquaporins, they're called aquaporins, aqua channels that let water across, because water can't cross the non-polar fatty acid chains. Water can't do that. It's polar. Water and oil don't mix. This is the oily part, the inside part. But you can have channels that have amino acids that face inside. The R groups of those amino acids are polar, and so water can make through. Ions, amino acids, glucose, so biomolecules, these will be also move across by gated channels, often by gated channels, and small molecules like oxygen, CO2, and ammonia will move, can actually squeeze through the lipid bilayer. They can move that way. So what we're moving is we're moving water, we're moving nutrients, and we're moving wastes in and out. Wastes out, obviously. So let's talk about what are the ways that things move across membranes. So there's three ways that things move across membranes. There's something called diffusion. We're going to talk about a lot, and it is it's like a thing you've got to wrap your head around, diffusion. But okay, diffusion. Then transport by protein, so facilitated transport or active transport. Those are these channels. They can either be passive or active. We will all talk about it. And then there's actual movement of the cell membrane. The membrane envelops things into vesicles, or vesicles fuse with the membrane and dump their contents out. So endocytosis and exocytosis are how things are called. So those are the three ways that things move: diffusion, protein assisted, and the whole membrane moving, enveloping. If you've ever seen an amoeba doing its job, you've seen endocytosis, it eating something. Okay, so let's talk about diffusion. This is there's a lot of vocabulary in diffusion, in the whole topic of diffusion, that we have to understand the concepts behind the vocabulary behind diffusion. So diffusion essentially is, there's the law of diffusion. Things follow the law of diffusion. Let me get the law of diffusion correct for you. What is the law of diffusion? The law of diffusion states that when there is a concentration gradient, okay, we'll figure out what that is. When there's a difference of concentration gradient, molecules will diffuse, will move to a region of lesser concentration. Okay? So let's back up a little bit. Let's define something. Let's define some terms here. Let's define the term solute and solvent. So those are the two parts of a solution, are solute and solvent. Solvent is the thing that a solute goes into. So for example, you have a thing of water and you plop some dye into that water. The water is the solute. The water is the solvent. The water is the solvent. The dye is the solute. It dissolves in the water. And basically, what defines solute and solvent is the overall amount. There is less of the dye, so it is the solute. It is dissolving in the water, and the water is the solvent. Now, if you reverse their amount, so all of a sudden there is more, you keep adding dye until there is more dye than there is what? There's more dye molecules than there are water molecules. Their definitions would flip. Then if there was more dye, the dye would become the solvent, and the water would become the solute. Now, in biological systems, what we're dealing with, water is almost always, I can say, I don't like saying always, but water is always the solvent, because the outside of the cell and the inside of the cell are always water. So we're always dissolving things in water. And this solute will be something else. Will be salt, will be any of the biomolecules. Any biomolecules will be oxygen, carbon dioxide. All of those things will be dissolved in water. It could be proteins dissolved in water. Those will be the solute, okay? Important. So, and then though the solute will move according to the law of diffusion. It will move from an area where it is greater concentrated to an area where it is less concentrated until those concentrations are equal. So what does that mean for us? I mean, well, let's gonna draw here. You'll be able to see it. So basically, imagine there's no membrane, there's no nothing, and you drop, it could be anything, some kind of solute inside water. What will happen is basically these things will spread apart until, like if we're in a box, they will spread apart until their concentration is equally spaced. That's diffusion. And the reason they do that is because of something called Brownian motion. All because everything has kinetic energy. Everything is temperature, is kinetic energy. So all these molecules, all the water that is in here, all the water molecules that are here, they're all vibrating against each other. And basically, they just spread all the things out. Now, this also means that if there's a membrane, things will move across a membrane, will diffuse across a membrane. So if there's a membrane and there is a greater concentration on this side of the membrane, and these things can pass through the membrane, eventually they will pass through the membrane until the concentrations are equaled out. Okay? Anything else I want to say about diffusion? We talked about the law. You can increase the rate of diffusion. That is one of the questions here. You can increase the rate of diffusion if you increase the kinetic energy. And you can increase kinetic energy two ways. You can heat something up, then the molecules will be vibrating and moving faster, so they will spread out faster. You can stir. If you stir, you drop some dye in a beaker of water. If you stir it, the dye will spread out and its concentration will become uniform throughout faster. That's one way. You can increase the concentration of the solute. So when you drop the solute in, if you, instead of dropping one drop, you drop two, there is more greater concentration of solute here, the diffusion will be faster because the concentration gradient from here to here will be greater. We're going to talk about concentration gradient in a second. You can decrease the size of the solute particles. Now, this is not practical. How are you going to decrease the size of molecules and stuff? But that would work, because what would happen is if you decrease the size of the molecules, they can fit in between the solvent molecules better and move faster. You can also decrease the solvent's viscosity. How are you going to do that? That's also not practical. And viscosity, what that means is how easily a solvent flows. So if you drop dye into honey, which has a very high viscosity, which means that it doesn't flow very well, that dye will never diffuse in the honey, or it will take like 1,000 years, 1,000,000 years. I don't know how long it would take for dye to diffuse in honey. But in water, which has a low viscosity, the dye will take a few minutes or minute, or it could be even seconds. Okay, so let's talk about now some other terms here. There is a very important term that are three terms: hypertonic, hypotonic, isotonic. Whew. These ones, students hate these terms, but okay, we've got our two sides of a membrane, and we've got membrane, and we've got some kind of solute. So in here, the concentration of solute is this, and here, the concentration of solute is this, okay? So there, and then, you know, maybe, and we've got water in here. Let's just make some water particles. Those are the solvent. Okay, so the ratios here are different. So there is a greater concentration of solute in here. There's more solute versus water. What that means is that this side is hypertonic, hyper, more, tonic, and tonic refers to the solute, than this side. That's what it means. Hypertonic to this side. This side has a decreased concentration of solute, and therefore it's got a lower concentration of solute, and so therefore this side is hypotonic to this side. Hyper, more tonic. Tonic refers to the solute. Hypo refers to less. Hypotonic, less solute. Hypertonic, more solute. See, it starts to get crazy. Okay? Now, if these had the same concentration of solute, they would be isotonic. Iso means the same in this scenario. Isotonic. Tonic refers to the concentration of solute. Hypertonic, hyper, more, greater concentration of solute. Hypo, less concentration of solute. Iso, equal concentration. So if I put a cell, a blood cell, let's say, into a hypertonic solution, that means that the cell is inside a solution where there is a greater concentration of solutes outside of the cell. The cell, the interior of the cell, is hypotonic to the exterior of the cell then. Okay? I know, we'll just go again. This goes gotta. Okay, so now, hypertonic. What else do I want to talk about? So we need to now talk about, so we talked about diffusion, we talked about isotonic, hypertonic. So let's talk about now, so we talked diffusion, and let's talk about osmosis. What is osmosis? Because you're going to be hearing a lot about osmosis, and you guys probably have heard osmosis. Often students answer osmosis on a lot of questions for things. What is osmosis? Well, osmosis is diffusion. Osmosis is diffusion, except it only refers to the movement of water. So it's osmosis is only the diffusion of water. Only water. Nothing else. No water only. It's water moving. That's osmosis. Osmo, when you hear osmo, water. You're thinking water. I'm just going to think about water. So, and if osmosis is diffusion, that means that osmosis follows the law of diffusion. Where there is a greater concentration of water, the water will diffuse to the area of lesser concentration. What are the implications of that? Okay, well, the implications of that are this for right now. So I've got a hypertonic side here and a hypotonic side here. Now imagine this membrane here that is separating the two sides is semi-permeable, and it doesn't let our big purple things through, our solute. It only lets water move through. So there is a concentration gradient going this way. There's a greater concentration of solute going this way, but it can't move across. There is an osmotic gradient. Osmo means water, going the other way, because there is a greater, the reverse is true here. There's a greater concentration of water now compared to this side. So there's an osmotic gradient going this way. And water will want to move across this semi-permeable membrane because it's following the law of diffusion. The law of diffusion is that things will move from greater concentration to lesser concentration. And there's an osmotic gradient here. Only water can move here. So water will want to move across this semi-permeable membrane. And as it moves, there are a couple things are happening. So there'll be an osmotic pressure on this membrane. We're pushing this membrane. That'll be osmotic pressure, just means the movement of water, the pushing of water. It's an osmotic gradient, osmotic pressure. So there's an osmotic gradient. Now, what will happen is that this movement of water will be to this side, and actually the level of water will drop here on this side of the membrane. More water will start to fill up this side until the concentration is equal. The ratio of water molecules to the purple molecules equals the ratio of purple molecules to water on this side. These could be anything. These could be proteins, sugar, salts, anything dissolved in the water. So what is this implication for cells then? So this could be a potential problem for cells, because cells there's water inside cells, there's water outside cells, and so cells have to control the concentration of solute inside the cells versus what's outside of the cells, because water will want to diffuse across these channels, across these aqua pores. So what cells can do is they'll have a limited number of these channels. They can have a limited number of these channels, so not to let a lot of water move through. Because imagine there is a greater concentration, so there's more purple things or whatever inside the cell than there is outside. Water will want to move in, and water will just keep moving into the cell until the concentration is equal. Well, cells have a limited finite volume, and the cell might burst. It's called deplasmolysis when that happens. We'll talk about that in a second. Okay, so osmosis, we've talked about that. Osmotic pressure is just the pressure of water moving across a membrane, the pushing, the crashing of the water against the membrane as it moves through. I hope I'm making sense out of this. So a concentration gradient is the gradient of solute, so not water, although it actually does, water does follow concentration gradient, but we don't call it a concentration gradient, we call it an osmotic gradient. And it is from greater concentration to lesser concentration. An osmotic gradient is from a greater concentration of water to a lesser concentration of water. Osmotic gradient, concentration gradient. There's also something called a pressure gradient. If you imagine these are gases, a pressure gradient just is the same as a concentration gradient, but it just refers to gases. So if there was a greater concentration of oxygen out here, there would be a pressure gradient for the oxygen to go into the area where there's a lesser concentration of oxygen. This is going to be important in respiration. There's going to be a pressure gradient where oxygen from inside your lungs moves into your blood. So let's talk about plasmolysis and deplasmolysis. Do I want to erase this yet? I really like these diagrams. I like them so much. Well, actually, is there an example of plasmolysis and deplasmolysis in... Oh, there is. It's this terrible hand drawing. Oh, that's not going to make any sense. I'll make another terrible hand drawing here. Here, we'll erase this, even though I like that drawing. Okay, so imagine a cell. Let's think about these things. So we're thinking about concentration gradients, osmotic gradients, osmotic pressure. So you've got a cell, and you put the cell into a hypertonic solution. So a hypertonic solution will be, there is a greater concentration of solute outside of the cell. So in the fluid outside of the cell, there's a greater concentration. So what's going to want to happen? And there is a lesser concentration in here. The inside of the cell is hypotonic. So what's going to want to happen is these things are going to want to enter the cell, but the cell membrane is semi-permeable. It might not let these things in. They might not be able to enter. There might not be proteins in the membrane to let them enter. So then what's going to happen is there's water in the cytoplasm. Those things might be able to enter or move, but there might not be enough proteins in the cell membrane to move them fast enough. So there's water inside and outside. Now, there's a greater concentration of water in here. It is hyperosmotic in the interior. If it's hypotonic, which refers to the solute, then it's hyperosmotic. That means there's a concentration gradient, an osmotic gradient of water wanting to move out of the cell. the cell will become dehydrated. That is called, just to make sure that I get it right, plasmolysis. Plasmolysis. And water will move out of the cell. All cells have water channels that it will move out of until the concentration is equalized. And what might happen is actually the cell might, what's called crenulate. And basically what that means is, this happens when you become dehydrated, when you don't drink enough water, is the cell will get fold up because there's not enough water inside the cell. It's like a balloon that's deflating. And water exerts something on the inside of cells called turgor pressure. Turgor pressure is the pressure of water pushing against the walls of a cell. And when there's not enough water, the turgor pressure drops and the cell membrane, it folds up. So this can happen to red blood cells. It makes it hard for them to carry oxygen this way when you become dehydrated. When you become dehydrated, the concentration of water in your extracellular fluid, in your plasma, becomes less. Your water is gone. You don't have enough water. And the solutes, the proteins, the salts, and all the other things that are floating around in your plasma, those are the solute. Their concentration becomes greater as a result because there's less water. So water starts to leave your cells. And that's called plasmolysis. Okay? Deplasmolysis is the reverse. So the reverse is, what happens is, you've got your cell. Let's make it a red blood cell, sure. And now you are in a hypotonic environment, which means you don't have, the concentration of solutes is very low in the outside, the extracellular fluid, the outside of the cell, and the concentration of stuff in here is greater. So now the concentration gradient is heading out of the cell, but the cell is a semi-permeable membrane. These proteins and things that are inside the cell might not be able to get them out fast enough or doesn't want to get them out. And there is an osmotic gradient. Osmotic, so refers to water, of water wanting to go into the cell to equalize the concentration gradient. What are the implications for the cell then? Well, water starts to enter the cell. And it goes into the cell, and the cell might not be fast enough pumping out the water, getting the water out. And what will happen is the cell will swell, the turgor pressure goes up, and the cell swells, and it pops and breaks open, and now is dead. This can happen. Dun dun. The Boston Marathon. How long has the Boston Marathon been around? I don't even know how long the Boston Marathon. I use this example so much. I'm gonna look up Boston Marathon. I don't want Boston Marathon bombing. Boston Marathon scheduled to take place 126 years of Boston marathons. Whoa. So over a century of Boston marathons. Now, for over a century of Boston marathons, at least to what are my knowledge, no one has ever died during the Boston Marathon from dehydration. So dehydration was that when the extracellular fluid is hypertonic, when there's a lot of solutes and so the cells shrivel up. No one has ever died from dehydration. But many people have died, one recently in 2018, from overhydrating, from drinking too much water. Because what happens when you drink too much water and you're running, you don't pee because you're running. I mean, sometimes you see the runners during marathons, they pee themselves. I've never run a marathon. I think marathons are, in my personal opinion, are not that healthy to run. I think they're really, especially for old guy like me, you guys, young folks, you got a lot of juice and stuff in between your joints. Old me, no. But what happens is when you drink a lot of water, you drink too much water, you wash out the salts and the solutes that are in your extracellular fluid. You can pee them out or you wash them out. The concentration of water in the extracellular fluid goes way up because you're drinking all this water. And so now you create a hypotonic environment for your cell. And your cell is now hypoosmotic, has a lower concentration of water, so water starts to enter the red blood cells. And the red blood cells burst, they deplasmolyse. And when do you need red blood cells to carry oxygen to your muscles? When you're running a marathon. And now your cells are bursting and they cannot carry oxygen, and they cannot carry oxygen to your brain, and you die. And so people die from overhydration. You know that whole thing, drink 8 cups of water a day and stuff? Pish on that. Your body will let you know when you will feel dehydrated. So, let's talk about some terms. So we have diffusion, which is the movement from a greater concentration to a lower concentration of solute in a solvent. Solute is the thing that's dissolved inside the solvent. Osmosis is diffusion for water. It's the movement of water from greater concentration to lower concentration, or the movement of water from a region of lower concentration of solute, because water is always the solvent, to a region of greater concentration of solute. So essentially, as the solute is dissolving, the water is moving the other way. It's osmosing the other way. As the solute is diffusing, water is osmosing. Osmotic gradient is the concentration gradient of water. So water will move from an area of greater osmotic concentration to lower osmotic concentration. Plasmolysis is when cells shrivel up because they've been placed in a hypertonic environment or a hypoosmotic environment. And deplasmolysis is when cells burst or fill up with water because they've been placed in a hypotonic environment or a hyperosmotic environment. You figure out what all that means. Okay, so those are the implications of diffusion of things moving across the cell. Okay, how much? That was 27 minutes. Oh no, we still have more. This is a huge, huge topic. I'm so sorry. We got to keep going. We're going to keep going. But you need to understand those terms, and you need to know what they mean. So here we go. Moving on. So we talked about diffusion, hypertonic, isotonic. What are the other ways? Because of deplasmolysis, blah, blah, blah. So how do things move across cell membranes? What are the capabilities? So first, things will move by diffusion. Those small molecules that I talked about will diffuse. They can diffuse through. So oxygen, carbon dioxide, ammonia. So these are not ions. They're not like salt, not Na+, K+, anything that's got a plus or a minus. Ionic things can't move across, but small molecules like O2, CO2, NH3 or NH4 can, okay? The other thing, all those other things move using proteins, these integral proteins. And they can move along the concentration gradient. So things can move along the concentration gradient. There's a greater concentration of something out here. It will move down this channel protein into the cell because it will try to it'll move around the concentration gradient. This doesn't require energy. It's free because of the law of diffusion. It will diffuse through this protein. And you can also close and open this protein, right? You can control if things move in. So glucose, for example, if you've got a lot of sugar outside of the cell, greater concentration of glucose outside of the cell, it needs to enter the cell via a integral protein. It will move across this membrane by diffusion. Now, sometimes for crazy reasons, you don't want a lot of glucose. You don't want the glucose coming in. So actually, there is a peripheral protein that sits on the side of the integral protein that controls the opening and closing of the glucose channel. And it is controlled by insulin. Some of you might have heard of diabetes. So insulin attaches itself, when there's the presence of insulin, it attaches itself to this peripheral protein, which then, when there's in the presence of insulin, it will open the glucose channel and glucose can enter cells. Glucose can be toxic to cells if there's too much of it. And then that glucose gets converted into energy. So when you need a lot of energy, your body will release insulin, or when you eat a lot of sugary stuff, your body will release insulin so that the glucose can enter the cells. So This is called facilitated transport. When you move along a concentration gradient into a cell or out of a cell across a protein, that is called facilitated transport. The protein, the integral protein, is facilitating the transport. So there's water channels, all kinds of proteins that let stuff through. Now, sometimes you want to move things against the concentration gradient. Hell yeah! And we can do that. Cells are awesome. We can move things across the concentration, against the concentration gradient. This is called active transport. So what happens is you've got proteins like this, and they're basically pumps. They change shape, they move, and they grab whatever it is inside and pump it out. So let's say there's too much sodium ions in here. We'll pump those ions against the concentration gradient. This requires ATP. It uses up energy moving against the concentration gradient. Maybe we want to pump ions in, potassium ions in. We have a pump to do that, okay? That's the other way. So you've got these integral proteins that can do facilitated transport, which is with the concentration gradient, doesn't require energy, or they can do active transport, different ones. They can do active transport, which requires energy, moves against the concentration gradient. Then, lastly, there is endocytosis and exocytosis. Let us look here. Just the best thing to do is to look at this. Endocytosis is, you've got, basically what happens is the entire cell membrane, so this is big things, big stuff, big things, or a lot of small things, a lot of small things or a lot of big things. The membrane, by using the cytoskeleton, so the cytoskeleton is that scaffolding that holds cells together. The cytoskeleton, it coordinates. The cytoskeleton is made out of proteins with secondary structure. Look, everything comes back. Everything that you learned before, everything comes back. You have to remember everything. You have to know everything. Those proteins, the cytoskeleton grabs onto the cell membrane and pulls it in, and whatever it is that's attached to the cell membrane will get sucked to a vesicle. That's endocytosis. There's two types of endocytosis. Phagocytosis. Phago is cell eating, so big chunks like another cell can be pulled in. Amoeba, phagocytose. Other amoeba or other small prokaryotes, they suck them in. Or big chunks of food, huge molecules like big glycogen or whatever, big sugar molecules, big things will be phagocytosed. And there's also pinocytosis, which is another kind of endocytosis, and that is where you suck in a lot of small things, or even water. You can even be sucking in fluids. So you would do this, but it'll be tiny, tiny things in here, but you just need a lot of them in. There could be like enzymes out here, tons and tons of enzymes that you want inside in a vesicle inside. Now, the flip side is called exocytosis. Exocytosis is just the absolute reverse. So you are a cell that makes digestive enzymes, and you are lining the duodenum in the small intestine, and you need to secrete a lot of these digestive enzymes all at once. What you're going to do is you're going to package these digestive enzymes into a vesicle. That vesicle is going to move and fuse with the outer cell membrane, and when it fuses with the cell membrane, it's going to dump all those digestive enzymes out into the duodenum, and they're going to digest whatever it is they need to digest, okay? So, how and what enters the cell? So how do things move? There's a nice table here in the notes. So we've got diffusion, which doesn't require energy. Things pass through the lipid bilayer. These will be small molecules with the concentration gradient. Small molecules, not ions. CO2, O2, NH3. Facilitated diffusion. Facilitated diffusion means you're moving through a protein with the concentration gradient. These will be things like glucose, water, amino acids, lipids, things like that you're going to want to move through. Biological molecules, proteins, either proteins and things like that. Active transport is against the concentration gradient. So that requires ATP. You're going to be using a protein, doing active transport. You're going to be pumping things out or pumping things in. These will be ions. Often this is like ions, sodium, potassium, things like that. and it's against the concentration gradient. And finally, you've got endo and exo cytosis. You've got two types of endocytosis, pinocytosis, phagocytosis, and you're bringing in proteins, large enzymes, disaccharides, large volumes of ions, things like that. You're moving against or with the concentration gradient. It actually doesn't matter. You are always going to be using energy because you're doing this cytoskeleton thing that requires energy to build the cytoskeleton and tear it down and all that sort of stuff. Microtubules and cell membrane coordinate to use cell membrane and vesicles to stuff out. That's how things move in and out of cell membranes. That's diffusion and osmotic pressure and all of that stuff in C2. We're done. That is a 35-minute lesson. I'm sorry, that's very long, but there's a lot in there. Do the review questions. They're really good.